Okay, should be set.
All right, just trying to find now the right screen. Oh my gosh. All right, can you guys see the slides or the presenter view?
Presenter.
Oh, man, okay. I know, we like just went over that. Okay. All right, so now you should have the slides,
Perfect.
correct? Okay,
Yeah.
awesome. All right, well, thanks, Adam, and thanks everyone for coming today. So as Adam mentioned, I'm going to be talking about eDNA tools for native species conservation, and in particular freshwater mussel conservation that the lab here at Columbia, Missouri, CERC, are developing. So to begin with, why use eDNA for freshwater mussel detection and management? eDNA has been shown to be a useful tool for species monitoring and surveying work, especially for species with low population sizes, those that live in difficult to sample habitats, species that may be difficult to identify in the field, and for systems in which managers have limited resources. And freshwater mussels are a good candidate for this eDNA survey work, as nearly 1/3 of all North American freshwater mussels are federally listed. Many have smaller declining populations, and traditional survey work can be difficult, being both time and labor intensive. For today's talk, I'm going to be discussing two different methodological approaches that we use in eDNA. And then I'll mention, or I'll talk about two projects that use one of these methods. So first, we have our targeted species-specific assays. Most of you know that these are going to be targeting just one species. These are associated with conventional or endpoint PCR, as well as quantitative methods such as qPCR and digital PCR. These quantitative methods allow you to quantify the original amount of DNA in the sample. And then we have community profile or metabarcoding assays, and with these we can target multiple species at once through the use of high throughput sequencing. I'm going to start off with a targeted species specific approach and just kind of have an overview of the entire process in general. So for instance, say we have this lake, and in this lake we have a number of species, and they're constantly shedding DNA into the environment. So we can take our eDNA sample. In that sample, we collect DNA and perhaps smaller organisms in their entirety as well. We can then extract that DNA. And then with the PCR, for our targeted approaches, we use species-specific primers and probes that bind to the DNA of just that one species of interest. That will therefore amplify, and our final PCR product is only composed of DNA from one species. We can then run that on an agarose gel to look for presence or absence of the DNA. Or, as I mentioned, we can use quantitative methods such as qPCR and digital PCR to quantify the actual starting amount. Finally, if we want to verify that that sequence belongs to the species of interest, we can use Sanger sequencing and verify that species. All right, so we're using this species-specific approach in a project that was funded by the Department of Defense's CERDIP program. And this is developing eDNA tools for quantifying freshwater mussel abundance, as well as monitoring breeding activity in different river systems. So the overall goals of this project are to gain a better idea of how eDNA moves in a system, and to inform about species presence, abundance, and breeding behavior. And we're using a quantitative PCR approach for this because we want to, in order to create this model, we need to measure concentrations of eDNA in a number of experiments, as well as in the field. And freshwater mussels make a great hopefully they make a great, we'll see, system for looking at eDNA detections in river systems, because the adults are for the most part sessile. They don't move as much as adults. And so we're really only concerned about getting that DNA that's moving downstream. So this is a diagram of sort of the overview of this project and how we're developing the model. So kind of at this top tier, we've got our biological inputs. And that's looking at shedding rates of eDNA, degradation rates of eDNA. So really the eDNA input into the system. We're also looking at environmental inputs, so factors like pH or temperature or water flow. Some of these factors may impact biological inputs like the shedding rates. And then we're also measuring hydrodynamic inputs of the two river systems. So we're describing the physical movement of particles, of eDNA particles in these two river systems. And so once we gather that information, our hydrology team will develop an eDNA transport model. And then finally, we will assess that model's findings based on field sampling that we're conducting. And so this is just sort of the same diagram, but just kind of showing in that middle picture there, the biological inputs. That's a picture of eDNA shedding experiment that we're doing with Mucket in the lab. And on the hydrodynamics input is a remote boat that's going out to measure various hydraulic and hydrodynamic inputs. So the majority of work has, for the most part, the biological and hydrodynamic inputs has been accomplished. So we've gotten eDNA shedding rates and degradation rates for 2 of our 4 species. The river systems have both been measured, and our hydrology guru, Brandon Sansom, is currently working on developing the transport models. A little bit of background on the system that we're developing these models for. So we're looking at 2 river systems, the Clinch River in Virginia, Tennessee, and the Big Piney in Missouri. And for each of those 2 rivers, we're developing or optimizing qPCR assays for 2 different species. And 2 of these are endangered. So the oyster mussel in the Clinch, or the spectacle case in the Big Piney. But we're also looking to understand eDNA detections of more common species, such as the kidney shell in the Clinch and the Mucket in the Big Piney. So as I mentioned previously, a lot of the lab studies for measuring eDNA shedding rates and degradation rates for the spectacle case in Mucket have been completed. And so I'm going to share some of those results with you all today. So we developed, we developed 3 of the 4 species assays. So again, those 4 species we need to develop qPCR assays for, one of which was already developed by UMESC for the spectacle case. And so we just tested it and optimized it, made sure it worked well in the lab. And then we ran a series of experiments. We ran eDNA degradation rate experiments for both the Mucket and the spectacle case. And I'm showing you some of the data we have for that. So we found that the half-life of Mucket eDNA is about 16 hours. And this is similar to the published degradation rates for other species. We also ran shedding experiments for Muckets, and we did 3 of these. We did a biomass shedding experiment where we had different treatments for different biomasses of Muckets in the aquaria, and we took eDNA samples throughout a 2-week period to look at eDNA shedding. And you can see figure 1 right there on the top right is showing the data for that. And we measured a eDNA shedding rate of about 550 copies per hour per gram for Mucket. I'd like to mention that that's actually a very low amount of DNA compared to data that we have for silver carp and big head carp, where they shed quite a lot more per gram. We also ran an experiment looking at how shedding rates are affected by feeding, and you can see on the bottom on figure 2, we found no effect of feeding on Muckets for eDNA shedding rates when we were running these in the lab. So we had a no food treatment, a low food, and a high food, and a high feeding treatment. We expected that since they're filter feeders, the more you feed them, the more they're probably going to produce eDNA, since they're more active and in the water column. We didn't really observe any sort of pattern with that. And we're also looking at temperature effects where we held mussels at different temperatures, and that's currently being analyzed. We were able to conduct similar experiments with spectacle case, and those data are being analyzed. And we're hoping to be able to conduct similar experiments with the other two species, the two Clinch River species, in coordination with Dr. Jess Jones. So moving on to the hydrodynamic inputs into this model. So here, our hydrology team led by Brandon Sansom and Mara Roberts, they've gone out and taken a number of measurements. I don't even know what all these measurements are, I gotta admit. I'm not the hydrologist, but they are. And so they characterize water depth, velocity, and water surface across a range of water discharges or river discharges in these two systems. This will allow them to develop and calibrate the models. And then they also were able to run last year, you'll see figure 5 on the bottom, where they did a dye release study on the Big Piney River in Missouri in 2021. It's just another way to try and calibrate and see how our eDNA transport model, how that works with a conservative dye tracer. And this work was done in coordination with Drs. Jennifer Tank and Ariel Sjogren, who also have a CERTA project related to eDNA transport. All right, so currently those models are being developed. From the lab studies, we'll be taking those shedding rates, and Brandon will be putting them into the models in order to try and come up with some sort of prediction about concentration of eDNA at various distances down the river from the mussel beds. So as I mentioned, the last kind of component of this transport model is to actually assess the models that the predictions of the models with actual field data. And so here I'm just kind of showing a diagram of what this kind of looks like. So we've got our mussel bed, and then we're sampling eDNA both upstream of the mussel bed, at the mussel bed, and several hundred meters downstream at various transects. And so we're basically sampling throughout the year. We're going to try and get about 6 different sampling events for each river. At each river, we have 3 different mussel beds, and at each mussel bed, we have about 4 to 5 different transects. So 100 meters upstream at the mussel bed, and again, 100 down to all the way up to about 4,500 meters downstream, depending on the mussel bed. So this is a sort of a diagram of what perhaps our eDNA data may look like. Hopefully getting no detections upstream, but of course we do have mussel beds upstream of other mussel beds, so that may not happen. We expect, higher concentrations at the mussel bed and subsequently lower concentrations as we move down. So let me grab a pointer. Here we go. So this is sort of a diagram of showing what we're kind of looking at here. We have eDNA on the y-axis and distance from the mussel bed on the x-axis. And so the green bars are the eDNA concentrations of our actual field samples. And then the colored lines represent 3 different model predictions based on different variables that go into those models. And so the idea here is that we want to compare our actual field eDNA concentrations with those predicted by the model and figure out which model is the best predictive. And in this case, model 3 best represents the actual field data. And we're hoping from this that we can gain better insight into the factors that affect eDNA transport. I want to talk a little bit about a second part of the CERTA project, which I think is pretty interesting. And this is looking at reproductive behavior with eDNA sampling specifically for freshwater mussels. So most of you may be aware that freshwater mussels are an interesting group. They have an interesting natural history in that many of them rely on a fish or salamander host for their young or their glaucidia to grow up before they finish and become adults. But the other really cool interesting life history trait or characteristic of freshwater mussels is that they have something called doubly uniparental inheritance. Most organisms have one mitochondrial genome, but freshwater mussels sometimes actually have two. They have a female mitotype and a male mitotype. And the female mitochondrial genome is found in female mussels in both their somatic tissue and germline tissue. The male and it's also found in the somatic tissue of males. However, the male mitotype DNA is only found in males, and it's only for the most part found in the germline, so they're gametes. And so we hypothesize that if we could design qPCR or assay-specific, qPCR-specific assays specific to the male mitotype, that we might be able to help measure when males actually spawn in the field. And since there's a lot of questions about, freshwater mussels are highly threatened and endangered, and they're having recruitment issues, are we even getting reproduction occurring in the field? So by designing assays that might be able to look specifically at the release of male gametes might give biologists some indication of what's going on in these populations. So here on the y-axis, I have the mitotype concentrations, and on the x-axis, I have time. This is sort of what a hypothetical situation might be like. So the majority of tissue is going to have that female mitotype. So that's what the majority of our eDNA is going to be. However, when a spawning event occurs and when males release their gametes, we might expect a spike in the male mitotype in our eDNA samples. And then females will then take in the sperm, fertilize their eggs, and then release their larvae, showing another but slightly maybe smaller peak of female mitotype for later on. And so we are designing or proposing to design species-specific qPCR assays for the 4 species male mitotypes. And so far we've designed male mitotype specific assays for the mucket and the spectacle case. We've tested, we're currently working on testing these assays with gonadal fluid extractions in female tissue samples. So far for the mucket, all male gonadal samples have amplified with the male mitotype assay. We did have one amplification from a female tissue sample, but I suspect that might be due to contamination. It's fairly difficult to get clean tissue, gametic tissue from a mussel, because it's right next to somatic tissue. And then we were able to hold spectacle case, endangered spectacle case here at CERC in this holding pond. And water flows in from this pond and it flows out. And we had them last year. And so we sampled during the period of time when we suspect that spectacle case may spawn. And those samples are being analyzed with our spectacle case male mitotype assay. So hopefully a lot more cool data to come with that. So moving on to eDNA metabarcoding and another freshwater mussel project we're involved with. So again, as I mentioned, this method allows you to sequence multiple species at once. And so the first few steps, collection, extraction, are the same, but it's at the PCR and sequencing steps where things differ. So instead of species-specific primers and probes, we have universal primers that bind to multiple species and amplify multiple species DNA. So our final PCR product is a multi-template product in which there's DNA from multiple species. And we can't use Sanger sequencing to sequence this type of DNA. And so we rely upon high throughput or next generation sequencing, which does allow you to sequence a multi-template sample. And so again, we can therefore look at our output reads and look at the database and see what species may be present based on the DNA that we're collecting. There's a lot of interest in developing eDNA metabarcoding assays for freshwater mussels. And this is what the objective was for this study. So specifically, we developed 2 metabarcoding assays that identify to species level freshwater mussel eDNA from water samples in the Clinch River. And these assays are currently being tested in other river systems. So again, a little bit of background. The Clinch River is in the Appalachian Valley in Virginia and Tennessee. It has the highest concentration of extant federally listed aquatic species, both freshwater fish and freshwater mussels. And in 1998, there was a large chemical spill leading to severe declines in populations of many different freshwater mussels. And so there's been a large effort in restoring these populations and reintroducing mussels back into these populations. And our collaborator, Jess Jones, was interested in the concept of perhaps using eDNA and eDNA metabarcoding to monitor this restoration effort. And so the first step in this project was to design metabarcoding assays that were hopefully specific to freshwater mussels. So we did that. We actually designed 2 different assays. The first one was based on a CO1. And it was actually based off of a primer set from Cho et al. In 2016, where they designed a number of species-specific primers for freshwater mussels. However, this one primer they found actually amplified multiple species, like over 20 species, I believe. And so we took that as our starting point because that's what we wanted. We wanted a primer set that would actually amplify multiple species. So we took that primer set and we compared it to the species of interest in the Clinch River. We added some degenerate bases to help improve its ability to amplify multiple species. And then we also developed a metabarcoding assay that targets the ND1 region. We then took multiple samples from the Clinch River and ran those with our assays. So results from this, the primers were developed from sequence data of 55 North American freshwater mussels across 29 genera. This just helps show that, or this just demonstrates that these assays were designed across a wide variety of unionid mussels. So we should hope and expect these to amplify not just a specific group, but hopefully unionid mussels across the entire group. We then tested these primers with freshwater mussel tissue samples from 30 different species, and they amplified all freshwater mussel tissue samples. We also tested them against 2 non-targets, Curbicula and silver carp. They did not amplify either non-target. And we also, from our sequencing data, we found very few off-target sequences. These all suggest that these unionid primers are specific to freshwater mussels. These, so they hope they should be freshwater mussel specific. And this is really important, especially for metabarcoding or universal primers, because trying to get them as specific as possible, but still amplifying all the target species really helps increase the assay sensitivity to your target, because these primers are therefore not being amplifying non-target DNA. So the non-target DNA is not competing with your target. Okay, and so I'm going to show you the results from this work, and a little busy, so I'll try and break it down for you. The first large column here are the sequencing results from our CO1 assay, and then from our ND1 assay. Across, we have the different sites on the Clinch where we took samples. In parentheses are the number of samples per site. So you'll notice at Indian Creek, we took 5 samples, and at Pendleton Island, we took 16. So there's fairly uneven sampling effort. And then on the Y, we have the different species that were detected with this assay. So overall, from our Clinch River water samples, we detected 19 different freshwater mussel species, 8 of which were federally endangered. and I highlight the federally endangered species with those that are in red text. If you look down here, we have the total number of species detected per site. One thing to notice is that our CO1 assay appears to detect slightly fewer species relative to our ND1. We were getting fairly even species diversity coverage with our ND1. However, the CO1 assay does detect some species that ND1 does not, such as the Epioblasma brevidens and Pleurobiema plenum. And so we suggest for folks who want to use these assays that it would probably be beneficial to use both. Kind of wrap it up here with these results. These assays are being tested by a number of other groups, including Jonah Ventures. Stephen Spear and Anna McKee and I are also working on a project using these assays with the Flint River drainage in southwestern Georgia. And we're here at CERC, we're also going to be testing these assays with two mid-Missouri rivers, the Borboose and again the Big Piney. And Willie Dokay and Wes Larson up at, they're currently now at the University of Alaska and NOAA, but they used a modified version of the CO1 assay and tested that in tributaries of Lake Michigan and the Grand. And they also detected about, I believe, around 19 different freshwater species in the Grand River using this assay. All right, so I'm going to just kind of wrap it up here. I put on here the different, as I mentioned, there's a lot of interest in freshwater mussel metabarcoding. So I've got our paper up here. These are the targets, the CO1 and ND1. But I also want to draw your attention to Prié et al., who in 2020 also came up and published an assay for freshwater mussels that targets the 16S region. And their paper was specifically designed for European freshwater mussels, but certainly will probably have utility for North American freshwater mussels. And then Coghlan et al. In 2021 also designed a 16S assay looking for eDNA from freshwater mussels in Canada. And so some future directions where I'm kind of wanting to see this work go, but also where we kind of need it to go. The CERDIP project that I mentioned, that eDNA transport models project, we're collecting hundreds and hundreds of samples, and we'll be using, again, those qPCR assays for our objectives. However, it would be great to actually also use the metabarcoding assays and look at seasonal changes. So for the CERDIP project, we're taking samples throughout the year, and hopefully with the metabarcoding, we can perhaps see some seasonal changes in freshwater mussel assemblages, or also just behavior and natural history of freshwater mussels, I think will become apparent from eDNA sampling. Next, let's see. Oh, yes. Definitely want to continue comparing the eDNA data that we get from these metabarcoding assays with freshwater mussel visual surveys. We did do that with our metabarcoding project in the clinch, but so I think we got about, I think the eDNA surveys got about 50% of the species on average that the visual surveys were finding. And so I'd like to try and do a little bit more diving into that and see if there's ways that we can improve. I think one way we can improve our eDNA data is by collecting larger volumes of water. So for the metabarcoding project, we were only collecting like 50 ml water samples. And then finally, a really important part is continuing to increase the genetic database for freshwater mussel species with both female and male mitotype data. There are just a handful of male mitotype sequences up on GenBank. I think they're all from CO1. It's really difficult to actually be able to get pure, just male gametic tissue. So, you know, I think this male mitotype assay is really interesting. I know others are interested in using it, not just in the field, but also in the lab where folks are raising mussels. But we really need to get more genetic information, as most of you are probably already aware of. There is a dearth of that for these studies. So I'm just going to finish it here. Happy to answer any questions, and of course acknowledgements. A whole bunch of people are involved in these projects. We've got our eDNA group, certainly our hydrology group, again, which is being Brandon is taking the lead on that, as is Mara. We couldn't do a lot of our work, especially with the endangered spectacle case, without our freshwater mussel husbandry group and our biologists for just helping us gain better insight into freshwater mussels and how their biology may impact eDNA results, of course, stats and modeling. And with that, I would be happy to take any questions.